My name is Shirley Curtis-Summers. I am a lecturer in Archaeological and Forensic Sciences at the University of Bradford. And this evening, I'll be presenting the isotope results from the Cold Case Whithorn project. So Adrian presented the first component a few weeks ago. So hopefully you caught his talk. If not, do check it out. It's online. So I'm very happy to be here with you this evening, and thank you all very much for joining us. So let's dive right in. Okay, so firstly, I'm just going to take a leaf out of Adrian's book and start with some thank yous. Firstly, thank you to Adrian and to Julia for inviting me on this project. It's a really exciting project, so I'm very honoured to be asked. So thank you very much. A special thank you to my research assistant, Orshi, for all your hard work on the isotope data. Thank you very much, Orshi. And thank you to the Cold Case team. There's a number of people behind the scenes been working really hard. So Isla, Bronwyn, Ruri, countless others, thank you all so much. And of course, this project would not be possible without the generous support and funding of a number of different bodies, and also support and collaboration from a number of different institutions and organisations as well. So thank you all very much. Okay, so let's start with the samples. Okay, for isotope analysis, we sampled 19 adults. Ages range from 17 to 45. They were 8 males, 6 females, and 5 were unknown sex. So not all were tested for isotopes. So for the dietary data, for example, 2 of the collagen failed, 2 individuals. So we haven't got dietary data for those. And some we haven't got strontium and oxygen data for either. So we didn't have all the teeth, or we didn't have some of the bone. Okay, but the majority we do have isotope data for. So we have 1 infant, just to get a kind of snapshot of infant diet, and later on we'll look at infant health as well. Now this infant was aged 0 to 3 years at death. That's a common age of mortality for the period. We find that at other sites as well. The osteologist who did the Whithorn collection, Amanda Cardy, suggested that there was a large number of perinatal and infant deaths in the early phase. So this is quite a common age for mortality. So it just gives us a nice snapshot of infant diet. Now we were restricted to the number of samples of fauna, so we just tried to select those that are obviously important to provide a faunal baseline. So we've got some herbivores, omnivore, and a marine. So we've got cattle, sheep and pig, sheep and goat, a pig, chicken, and a shark. Come back onto that. So because we've only got a small sample number, we have to use established faunal data, just to support that faunal baseline. So the data we'll be using for that is from Port Mahomet, which is contemporary, but not contemporaneous, sorry, but not geographically identical, obviously. So over in the northeast highlands of Scotland, but nevertheless it provides a good faunal baseline. And we also are using faunal data from later medieval Whithorn, which is a study that was done from the Fayfield a few years ago by Gundula Mulder, Janet Montgomery, and colleagues. So we're using that data as well, which will prove really useful. Okay, so you may recognise this burial plan from Adrian's talk a few weeks ago. So the ones that are coloured are those that we have analysed. And the majority of these are radiocarbon dated to the 7th to 8th century. These 2 black ones, as Adrian mentioned, are the ones that failed. So no radiocarbon data for those and no dietary data for those, but we do have the teeth isotope data. So we have the mobility data, just not the diet. This individual up here, buried just outside the chapel wall, is quite interesting. So we're going to come back to that individual a few times. So just bear this one in mind, buried in a log coffin just outside the chapel wall. Now, what's interesting here, and we'll touch back on this a bit later as well, is we have 3 individuals, all of whom had TB. So we have these 2 individuals here and this one up here. I hope you can see my cursor just moving around a bit. So they had TB, highly infectious disease. We're going to touch on that a bit more later as well. And they're from the 8th century, potentially monastic phase that Adrian talked about. And then we have our infant in the top right-hand corner here from the children's burial cemetery. Okay, so I'm going to start with the isotope results, and I'm just going to give those of you that are not familiar with isotopes a quick run through, just an overview, and then we'll move on to the results again. Okay. Right, so the elements that we're using for isotope analysis are carbon, nitrogen, sulfur, and oxygen. Now, the carbon, nitrogen, and sulfur are from the bone, and that gives the adult signature. Okay, and then from the teeth, we have the strontium and oxygen, and that gives the childhood signature because the teeth develop during childhood. So the isotopes used in this study are present in the biosphere and are incorporated into human and animal tissue via the ingestion of food and water. So depending on the isotope analysed, we can reconstruct the types of foods consumed and or patterns of mobility. So for example, we have carbon and nitrogen and sulfur that can be ingested through the plants, and then the animals that eat the plants, and then in turn the humans that eat the animals. So those elements get incorporated into our tissues. And the same with oxygen from rainwater into rivers and lakes and so on and so forth. Local water sources, ingesting from those and taking up that oxygen isotope into the tissue. And the same with strontium, that's a geological source, and that's incorporated into the bedrock and then absorbed into the plants and again through the food chain. Now, there's a number of factors that may affect this. Isotope analysis can be affected by such physiological stress, for example, so that can elevate nitrogen values. Food preparation practices such as boiling or stewing or brewing, that can alter oxygen isotope values. Other feeding practices such as breastfeeding, that can alter nitrogen and oxygen values. And location from the coast to the coast, for example, that can alter sulfur and strontium values. So the usage of a multi-isotope analysis alongside the archaeological and, where possible, historical evidence is obviously of vital importance. So this is just a diagram to give an idea of what an isotope plot looks like. So I'll just quickly run through this. Basically, carbon and nitrogen can distinguish between marine and terrestrial consumption. So marine ecosystems have a much larger food chain than terrestrial ecosystems. And this results in aquatic organisms having higher carbon and nitrogen values. So higher nitrogen going up here, higher carbon going along to the right. Okay, so more enriched in carbon. Nitrogen can also determine trophic levels, and this increases around 3 to 5 parts per mil in nitrogen, each step up the food chain. So for example, these are all the nitrogen values, and if a omnivore came and ate a herbivore, it'd take on that nitrogen enrichment to be higher in the food chain. If a human came along and ate a herbivore, it'd take on that nitrogen enrichment, and so on and so forth. Carbon can distinguish between C3 and C4 plant consumption. So what I mean by that is C3 plants are those that are native to Europe, obviously including Britain. And they include plants such as barley and oats and wheat. So this is the C3 area that we would expect to see C3 plant consumption. Over here, we see the C4 plant consumption, and this is from more tropical zones such as the Americas, and that includes plants, plant foods such as millet or sugarcane. Sugar's a really interesting one because prior to the late medieval period in Britain, so sugarcane is not a native plant to Britain, so prior to the late medieval, you would not expect to see that C4 component in the diets of humans. Okay. Then after the late medieval period, it becomes more prevalent. So it's only really for high status elite tables in the late medieval period. Thereafter, it becomes widespread. Before, in our period, for example, early medieval Scotland, you would not expect to see that C4 component. Okay, so basically terrestrial C3 over here, marine C4 over here. Higher up in the food chain for terrestrial, higher even still for marine, and freshwater fish can pretty much go all over the place depending on the type of freshwater fish because they migrate from freshwater to marine and back again, so on and so forth. Hopefully that makes sense. We'll move on to the results now. Okay, so these are the carbon and nitrogen results from early medieval Whithorn. And what we've done here is, as I said earlier, is use some established faunal isotope data for our faunal baseline. Okay, so we have a range of fauna here. We have the herbivores, so we've got cattle, sheep, we've got the deer down here. So we've got the herbivores, then we move up to the omnivores. So you see that stepwise enrichment, you see that trophic level enrichment as they move up the food chain. So we have omnivores, we have a dog over here, we have pig, we even have a chicken. So these would expect to be a similar diet to the humans. These are the humans here, this cluster, because they would have been fed human food waste. So you would expect them to have a similar diet. This line here represents 100% terrestrial diet. So anything within this area is predominantly terrestrial diet. As we go over here, we get a mixed diet, and then here is a 100% marine diet. Okay, so the majority of Whithorn humans are within the terrestrial range. And as you can see, they're a trophic level above the majority of the herbivores. And this suggests that they're a trophic level above, so they're consuming these animals, okay? Obviously not they're probably consuming the pig, but the pig is being fed similar types of foods to the humans. That's why it's higher, as in the dog as well. And then we have our marine/freshwater. I've put marine/freshwater because these are char. These are from Port Mahomet. And char is an anadromous fish. So it migrates from freshwater to marine freshwater. So its isotope values can mimic either terrestrial or marine. So it really depends on the freshwater fish that you analyze. And then we have our marine sample, mean sample here for marine from Port Mahomet. So higher in the food chain, higher in nitrogen and more enriched in carbon, and our one marine sample from this study here. So you can see why we need other established data to be able to help our faunal baseline. So this all looks a bit busy, so we need to look at the humans in a bit more detail. So we'll move down. And these are just the humans now with the animals taken out, just so we can have a bit of a closer look. So these are humans that are separated by sex, so male, female, and those that are unknown. And as we see here, the majority of humans are within the same trophic level. So they're within 3 to 5 parts per mil nitrogen, and they're all fairly similar in carbon as well. We've seen a little bit of variation in carbon, especially from this individual here. So this is a male, aged 25 to 35 at death, buried in the burial chapel phase. If you're not sure what I mean by that, go and check out Adrian's talk. So it's the later burial chapel phase. slightly higher carbon, although not, vastly within the marine range. Possibly some fish consumption. Unfortunately, we have no strontium and oxygen for this individual, but the sulfur might tell us something. So we'll have a look at that in a moment. And then we have our infant up here. So as you can see, the infant has the highest nitrogen of all the adults. Not really a trophic level above the adults, however, just on the cusp of it there. Now, we expect to see infants at this age with high nitrogen because they're essentially consuming their mother's tissue by breastfeeding. So they're usually a trophic level above the mother, at least, or the humans, the rest of the adults. Now, what we may be seeing here is the start of the weaning process. So when the child starts to be taken off breast milk and start to be weaned, their nitrogen levels drop more in line with the adults. Now, another reason could be some form of physiological stress, and that's known to elevate nitrogen values also. So we can either be seeing some form of weaning pattern or some physiological stress in this child. And we need to do further analysis on this child, so watch this space on that. So what we're seeing is a fairly homogeneous diet, slightly enriched carbon for that individual and enriched nitrogen for the infant, but predominantly terrestrial-based diet. Okay. So what I've done here is just plotted the same individuals, but by the burial phase. And again, this was explained in Adrian's talk. So if we look at the data by burial phase, we see the earliest 7th century and the 8th century, potentially monastic phase, clustered together. So they've got similar diets. So they're the crosses for the 7th century earlier ones. There's 2 crosses just hiding in there, and the yellow diamonds. So they're the ones that kind of cluster there, similar in carbon, little bit of variation in nitrogen, but it's still within the same trophic level. When we get to the later burial chapel phase, we see it's only 3 individuals, but we do see some more variety and a bit more of a spread in both carbon and nitrogen. And again, this is the one that we looked at before with the previous plot and suggested it possibly fishy. Again, we'll see what the sulfur says about that. So it seems that the later burial chapel phase may have had a more mixed diet than the earlier 7th and 8th century. So that's interesting in itself, and that's something that we can look at in more detail hereafter. Okay, so I just thought it'd be interesting to look at some site comparisons as well for the carbon and nitrogen. So I've just selected a few sites and some contemporaneous sites and some slightly later as well. So we see that early medieval Whithorn has a similar mean average value to those from St. Patrick's Chapel in Wales, from Port Mahomet in North East Highlands, and Cramond in Edinburgh. What else have we got? Oh, and interestingly, it's very similar. So this is the Whithorn one here, our circle, our early medieval Whithorn, and it's very similar to the red diamond here, which is the later medieval Whithorn lay group from the Fayfield study a few years ago. Now this is different from the clerics from the same study, so they're more enriched in carbon and nitrogen. And the author suggested that they were consuming fish. These high status clerics were consuming fish. And this is in stark contrast to the monastic site over at Owen Bristy that was suggested to have a much more austere diet. So it's interesting that we just see these different sites. The majority are homogeneous diet, which we see across the board. A number of sites in Scotland and England. We've got a bit of variation in Ireland, but there's other Irish sites as well that are similar. Okay. Okay, so moving on to the carbon and strontium isotopes. So what we're seeing here is, these are all the humans again, and we've got our terrestrial mean here, quite high in sulfur. So this is sulfur now we're looking at, so quite high in sulfur. This is a marine enrichment, and this is most likely due to consuming animals, so the humans, consuming animals that were fed on foods treated with seaweed as fodder, for example, and that's where the marine enrichment is coming from. It's not coming from them eating fish. Now, the variation between the humans may suggest some variation in the distance of habitation from the sea. And we'll come back to that with the mobility results. Now, you'll notice this one has a very low sulfur value, and this is our outlier, our individual buried in the log coffin outside the chapel wall. Very low in sulfur from the 8th century monastic phase. Also, this individual was quite low in nitrogen as well, and it suggests that this individual was consuming foods further inland, so habitation was not near the coast. And we're going to come back to this individual later with the mobility results. Again, we've got this one that we suggested was a bit fishy, slightly higher carbon than the majority, but the sulfur is kind of tying in with a number of other individuals as well. So again, it's just suggests that it's not necessarily fish consumption, but affected by uptake from animals that were fed on marine fodder, marine treated fodder, for example. Okay, our one marine sample of our shark is, as would be expected, quite high in sulfur. So this is, we originally thought this was a cod, and it turned out to be a shark. It's not the shark you're thinking of. The early medieval people at Whithorn weren't, you know, consuming great whites every day of the week. It's actually a taupe shark. And taupe sharks are an inshore shark, commonly found in British coastal waters, including Scotland, still fished today. And this, I think, is a similar size to cod. So it's not one of those huge, big sharks that you would imagine, as with this one here. So what I'm saying is that they weren't consuming shark every day of the week. We don't know what the use of shark was for. It could have been for other uses other than consuming this type of fish. There weren't very few fish bones found during this phase, and we're going to come on to that later as well. I would like to thank Hannah Russ for identifying this as a taupe shark. Thank you, Hannah, because it was originally thought to have been a cod. So thank you very much. Okay, so that's the carbon and sulfur. We'll look at some site comparisons again, just very briefly. As you can see, we've got the terrestrial fauna here from Whithorn, and we're looking at St. Patrick's Chapel also, and also from Navan Fort. Now, Navan Fort is a much earlier site, early Iron Age, I think. But it's an interesting one to include because it's been described as a ceremonial feasting centre. And this is mostly due from the large number of pig remains. And the author stated that these samples are unusually high in sulfur compared to other sites in Britain. However, as you can see here, we've got St. Patrick's in Wales, and we've got our samples from Whithorn that have similar carbon and sulfur values. And these are both coastal as well. So it's interesting to compare those. And then when we look at the humans, similarly, we have Whithorn and St. Patrick's Chapel, very similar in both carbon and sulfur. Again, due to coastal proximity, we've got Cramond, which is a bit lower in carbon and sulfur than the previous. And the author suggests that this is from a terrestrial diet. Okay, and not all were local potentially. So that's all she studied. So I'll leave it there for her to reveal more at some point. Then we look at Landau. This is a study from Katie Hamer as well as St. Patrick's Chapel, and this is very low in sulfur. And the author suggests there that this may reflect individuals who consumed foods that from the hinterland, so away from the coast. So you can just see the comparison there in terms of the variation in sulfur. Okay. So what does this all mean? Well, let's look at the archaeological evidence first. So from the faunal remains, there were very few fish bones found in these early periods. We have that one shark. I think there was only one shark bone from period 1 and 2 from period 2, for example. So we obviously need to sample more marine to get an idea of what's going on here. There were very few very few sheep also from period 1, so from the 7th century. And this is a similarly low incidence found at other sites in Scotland, Ireland and Wales. But you tend to find the opposite at Anglo-Saxon England. It tends to be more sheep than cattle or pig. Now at Whithorn, this increased in the Northumbrian phase, and that was also paralleled in Ireland. So, for example, we have in the 7th century, cattle dominate, dairying is really important, and then thereafter it's prevalence of pig and then sheep, so not many sheep bones. Then in the 8th to 9th century, cattle are still dominating, but we see the opposite with prevalence with the other two groups. So we see more sheep instead of pig this time. And then the midden, we have a midden that's described as high status. That suggests animals such as lamb and suckling pig. There's a high incidence of roe deer in both period 1 and 2. And that's suggested to be superior to red deer meat. So it seems that's a high status foodstuff as well. So the isotopes tell us that early medieval Whithorn had a predominantly terrestrial diet, but when combined with the archaeological evidence, such as the glassware and the pottery also, and the faunal evidence, it suggests that this terrestrial diet was no means poor, with animals consumed that may reflect high status feasting. So we have a lot of lamb, pig, venison. So really, you know, high status cuts of meat and types of food. Okay, so let's move on to the mobility data. So that's the dietary data, and let's move on to the mobility. And this map shows the strontium and oxygen values across Scotland. So the strontium are the colours, so there are different strontium values. And then these dashed lines are the oxygen rainfall values. So as you can see, all the different colours, this shows the complex geology in Scotland. So the identification of a non-local place of origin, i.e. stating where somebody may have originated, this should always be considered tentative at best. And this is due to the presence of geological formations of similar characteristics throughout the UK and Europe. So for example, we have strontium values up in Caithness, for example, could be similar to down in the borders and even down in Yorkshire and Wales. Okay, so we normally need to combine that with oxygen to try and refine that data. Oh, sorry. Okay, so let's look at the strontium results first. And what we have here is of the 15 adults sampled, we potentially have 8 that are local to Whithorn. That's this group here. And they're a mixture of male and female, as you can see. And we have 6 that are potentially non-local. They could possibly still be within the Galloway area, but further inland and possibly further east as well. And then we have our one outlier again, that one that was buried outside the chapel wall in the log coffin with low nitrogen and low sulfur, very high strontium. So didn't grow up in Whithorn and probably not in Galloway either. And remember, low sulfur, so not local to the Maccas later in life either. So that individual, remember, was 17 to 25 at death. So it's potentially that he arrived in the area during early adulthood. Okay. We can't say whether he died shortly thereafter, but certainly wasn't born or raised in the area. And then we have another individual, this female at the very bottom here, so the lowest strontium value, just on the edge of the local range. So it's this individual was consistent with a coastal location, but again, throwing some caveats in here. It's also consistent with geology that can be found across other parts of southern Scotland and England as well. Now, the carbon and nitrogen for this female does not, as with most of the adults, does not suggest a marine diet. So again, that marine signal could be due to eaten animals that had that marine signal in them from their fodder, for example. So seaweed such as kelp, that has a very high strontium content, and it was used by many island and coastal communities as fodder in Scotland. So it's understandable that you would see this incorporated into human tissue from the animals. Okay, so what I've done here is just plotted in the later medieval Whithorn data from Golden and Mulder's study. And what we see here is that the later medieval clerics and the lay, so they're the later medieval clerics and they're the lay group, are plotted there. So most of the lay were local. Most of the later medieval lay were local, as we see with our study, the early medieval group. But the majority of the clerics were non-local here. These are non-local, apart from this one individual here. And they did not grow up in Whithorn. And we have one outlier here from that study. We'll come back onto that later. Okay, so in order to better include or exclude individuals as being local, we'll need to compare the strontium to the oxygen and see what patterns are coming out there as well. Okay, so this is the strontium and oxygen results. And what we're seeing here is that some individuals appear to have a colder northeasterly origin. And then potentially we have a few with a warmer western origin. Now, the drinking water values that are not plotted here, but they appear to correlate with this east-west spread. Okay, so we have locals, but we have some that are potentially more east origin and some slightly more west. Again, we have our outlier here, origin of further inland, possibly eastern or northeastern highlands of Scotland. Now, I'm going to be a party pooper again here and just throw in this caveat to state, to remind you that practices such as boiling or brewing or stewing foods can result in higher oxygen values and place an individual in a warmer climate, i.e. from the west, than they originated from. So some of these individuals that may have a warmer isotopic value could potentially actually be from the east. So you can see the complexities of the mobility studies and how we need to kind of build a picture as much as possible to glean the place of origin, although we can't pinpoint the place of origin. So all we can say is this individual was further inland towards the east and towards northeastern highlands potentially. Okay, so let's just go back to the later medieval Whithorn individuals as well and see where they plot with the strontium and oxygen. So the blue are the lay group from later medieval Whithorn, from the Fayfield, remember, and the crosses are the high status clerics. So the authors of that study suggested that the clerics grew up further east or north. And we have this one later medieval cleric here as the outlier. He was identified as Henry, who was Bishop of Whithorn between 1253 and 1293, and suggested to potentially originate from the southern uplands of Scotland. Whereas our individual here is potentially much further east into the Highlands, but not coastal. So you can see the difference between the later medieval bishop outlier and our earlier medieval outlier. So there's quite a difference there. So it's interesting just to see other studies. And this earlier study from later medieval Whithorn was vital for us to be able to build on top of that data and have that baseline data to be able to match against. So what we're doing now is we're just trying to build up that isotopic data because it's really sparse in some places in Scotland, and we really need to do more analysis to make that data more robust and make our interpretations more robust. Okay, so what I've done here is just gone back to the burial plan and just outlined those that are potentially local and non-local. So all the locals and non-locals are spread across different burial phases. And again, this is covered in Adrian's talk. So those that are within the red hatched squares are local to Whithorn. These two far ones here, the two orange ones, are plank coffins. So they're the earliest 7th century burials. All of the locals are were buried in log coffins in the 7th to 8th century. And our outlier here, again, more from the eastern highlands of Scotland, still buried in a log coffin, very prestigious burial, yet outside the chapel wall. That's quite intriguing as to why that could have possibly been. And I've just realised that this one here, good job I spotted it. This one is actually non-local, not local. So I've covered it in red. Let's pretend that's a blue hatched square. So that's a non-local, that's our non-local outlier. Okay, so good job I found that. So non-local outlier, all the other red ones are local. Right. Okay, then we have the blue that are non-local to Whithorn, as our outlier is. Some of them may be local further north in Galloway, or further inland east. Okay, one was buried in a stone kist. I think it is this one, yeah, stone kist burial. And then we have one that is buried in a coffin. I think that is this one, yes, and I think the rest are log coffins again, or the majority of them are. So it's interesting that potentially we have locals and non-locals buried together, buried alongside each other, I should say, sorry, apart from our outlier. And he seems to be he, sorry, don't know if it's a he or a she. this individual seems to be an outlier in more ways than one, in position of burial and location of origin. Still a prestigious burial nonetheless. And just as a reminder, we have 2 non-locals and 1 local that were all buried in log coffins and all had TB. And they are from the 8th century monastic phase. So that's quite interesting. So we have these 2 individuals and this individual. So 2 non-locals and 1 local, buried in high status coffin, all had TB, highly infectious disease from the potentially monastic phase. So yes, so that's quite interesting. So that's something I'd like to look into in more detail. And if anybody has any similar results from their study, do let me know. Okay, so what does this all tell us? Well, the 5th to 7th century has been described as the age of migration, and this is due to large-scale movement of people after the decline of the Roman Empire. Some scholars have argued for strong contacts between Whithorn and Ireland and noted parallels of log coffins and stones. And of course, the links with Gaul and the Mediterranean from the glassware and the pottery. And this points to evidence of wide trade links. As Adrian said in his talk, there's not much evidence to suggest large waves of migration from Ireland. So, if indeed Whithorn was an elite Christian centre at this time, could we be seeing evidence of some movement of high status people from the east and settling in Whithorn? Now, was this movement due to trade, political alliances, or simply for pilgrimage? Which was obviously encouraged by the Kingdom of Northumbria at the time. I say obviously, who knows, but potentially it was influenced by the Kingdom of Northumbria. So could our 8th century monastic outlier from the east, for example, be a high status pilgrim who possibly sought healing shortly before death? Hopefully you'll forgive me for venturing into the realms of speculation here. But until we can close the gaps in our knowledge, and there are still many gaps, this serves as one theory for now. And I'd appreciate your feedback on this as well. So there's much more work to be done, and these few individuals represent a mere snapshot of life at early medieval Whithorn. So, in summary, we have predominantly terrestrial-based diet, which is the norm at the time, although there is some variation at some sites across Scotland, but we do see a predominantly terrestrial-based diet during this period. However, evidence of high-status foods that were being consumed. We have a number of locals to Whithorn, males and females from the 7th and 8th century burials. There's evidence, established evidence from that of material culture links between Ireland, Gaul and the Mediterranean through the pottery and the glassware, for example. But the isotopes are telling us that there are a number of non-locals that are coming from the east. Okay, so it's, as we said before, it's not a strong connection with Ireland in terms of mobility. It seems to be they're coming from the east, either whether that's due to alliances or pilgrimage, who knows. But it gives us a good indicator of people moving across Scotland to Whithorn to either settle or visit for pilgrimage. And what we need to do now is, excuse me, we need to do more analysis and build up this data to get a clearer picture. Okay, so that's the isotope component, and I just wanted to end on the public engagement component, because that's very important as well. So this is really important because we need to share our findings with the public and engage them in a number of ways. And we've done that over the last couple of years at some really fantastic events. We've done a number of school workshops with the children. We've done public events such as the International Skiffy Worlds event in Stranraer, which is hugely successful. Focus lectures such as we are presenting to you now, and obviously media outputs as well to engage wider audiences. And the local communities of Whithorn and Stranraer have been really fantastic in engaging with us on this project, and we hope this relationship does continue further. So a big thank you to any locals that are listening this evening. So where next? Well, I'm sure it's evident from this talk that we need to do more scientific analysis. There are gaps in our data, there are gaps in our understanding. So we need to do more analysis to broaden our understanding of early medieval Whithorn. We need to do some key investigations. For example, looking into child diet and health. We've only seen one infant this evening, the results of. So we need a broader picture on that. Also building some osteobiographies. So looking at the life ways of people from birth to death. And there's a number of ways that we can do this. And that's something we're working on or planning at the moment. Obviously more public engagement in events, which are hugely important in engaging local communities and wider audiences. And of course, we need to understand the broader context to contribute and expand our knowledge of past lifeways in medieval Scotland overall. And this is hopefully through outputs such as this and collaborations hereafter. So it's not just the cradle of Christianity that we're focusing on now, and this is a term that's very much linked to Whithorn, but we now turn in our attentions to the lives of the people themselves, from their bodies, essentially. So in essence, we're moving from the cradle to the grave, so to speak, if you pardon my play on words. And I'll leave you with this quote from Professor Charles Thomas. He opened this at the very first Whithorn lecture in 1992, and it reads, "The poetry of history lies in the quasi-miraculous fact that once on this earth, once on this familiar spot of ground, walked other men and women, as actual as we are today, thinking their own thoughts, swayed by their own passions, but now all gone." Hopefully, the work at Whithorn ensures that although they may be gone, their stories in some way live on. So I just want to say thank you very much for listening this evening, and I look forward to our Q&A.